Well, I'm gonna start. I'm gonna start introducing Xiangyu. First I'm gonna say thank you all for coming here this Friday. I'm surprised in a nice way, and it was kind of unexpected to see so many people. So thanks a lot for that. And I would like to introduce, as I said, Xiangyu. He's now a postdoc working with Eric Allan at the University of Bern in Switzerland. And he's working with Eric and with me in things related to coexistence mechanisms, functioning, and how we can relate the dynamics of the populations to the many functions that we observe in the communities, such as community biomass production, pest control, mycorrhiza, etc., etc. Xiangyu, he did in Leiden his PhD in the Netherlands at the university that study above ground and below ground processes, and he's bringing some of his expertise into what he's doing right now. I am really happy that Xiangyu is going to be here till the end of February, so if you want to talk with him, he will be delighted to do so, and actually encourage that. He's in the green building, and we know that the red building and the green building looks like Sevilla and Betis, and hopefully this divide, I don't know how to solve that, but some interconnection will be desired. So Xiangyu, thanks a lot for the presentation. You don't need the micro, right? You do have one. Okay, thanks a lot for coming here.
Thanks, Oscar. Thanks for the very nice introduction. And hello everyone, I'm Xiangyu. And today I'm going to share my latest work during my postdoc about linking species coexistence and diversity effects on ecosystem functioning. And as many of us already know, that biodiversity can lead to higher ecosystem functioning. So from this figure, you can see that the higher diversity of plant species will lead to higher above ground biomass production. And this pattern even intensifies over the years. So this has been often reported. And later, ecologists, they expand this framework to multiple functions in the ecosystem or plant communities. And here is the effective size of diversity. And you find that the majority of the effects of diversity are positive on different or multiple ecosystem functions. So here the information is that biodiversity enhances ecosystem functioning. And the effect of global change factors can greatly influence ecosystem functioning directly, and it can also indirectly influence ecosystem functioning by influence biodiversity, such as reduce or cause the biodiversity loss. It can also greatly influence the community composition and then indirectly influence ecosystem functioning. And this is mainly through the influence on functional composition by shifting the community functional composition to the fast end, which means the fast-growing species start to dominating the community in response to some global change factors. For example, land use intensity increase the dominance of fast-growing species, which will indirectly influence ecosystem functioning. And this has been shown by Eric Allan, who is also my team leader in Switzerland in our group. So, regarding the underlying mechanisms of these positive diversity effects on functioning, there are two main ecological processes or high-level processes or underlying mechanisms, what we say. And one is the complementarity effects, which means that species in diverse communities, these species are more niche differentiated. So they lead to higher complementarity by complementarity to use the resource, for example. And this can lead to positive diversity effects. And also, if a community dominated by a few high-performing species by outcompeting the inferior species, and this process can lead to higher selection effects, and this can also lead to higher functioning. And actually, these two high-level processes, they well connected with species coexistence mechanisms, However, these two are often studied separately, so they are separated as two different research themes. And in biodiversity ecosystem functioning, people often explain the complementarity effects via higher niche differences, or explain the selection effects by fitness differences or competitive asymmetry, but they are rarely connected. So these two frameworks. And especially in multi-species communities, this is a challenge. And based what I have introduced just now, we would expect that higher niche differences in plant communities will lead to higher complementarity effects, while higher competitive ability difference or fitness difference will lead to selection effects dominating by a few species. And in multi-species settings, there are other important interactions happen. For example, facilitation and indirect interactions. And for indirect interactions is the case like the intransitive interaction I show here, rock, paper, scissor system. So only 3 species they present together, these 3 species are able to coexist, but each of 2 they cannot. And follow the theory, we would expect that in multi-species communities, indirect interaction actually they can increase the opportunity of coexistence by leading larger niche differences, and then maybe indirectly influence complementarity effects. And it can also directly influence the diversity effects, which is complementarity and selection effects. But we actually don't know. There is no study yet to link the indirect interaction with the biodiversity effects. And as I mentioned earlier, functional traits in plant community are also very important. And first is the fast-slow traits or functional composition. The larger values of functional composition here indicated by community weighted mean of specific leaf area. So the larger values means the community are more towards to dominated by fast-growing species. And the lower values of functional composition means communities are more dominated by slow-growing species. And the slow-growing species, they are more adapted to low resource conditions. Therefore, we expect they are more niche differentiated than fast ones. So by saying this, we would expect that fast communities, they would have lower niche differences, and they would have higher competitive ability difference. Slow-growing communities or communities dominated by slow species will be opposite. And also, we have another index, which is functional diversity. And functional diversity can indicate how species are differentiated in their niches. So we would expect that functional diversity lead to higher niche differences. And these relationships or this framework could also depend on global change factors. And the previous studies have shown that nitrogen actually can reduce complementarity effects and increase selection effects. And this effect could throw changes on the coexistence mechanisms. For example, so nitrogen typically thinks we adding nitrogen to the plant community, we can knock out one of the dimension because we added enough nitrogen to the system. The species, because before they are niche differentiated in this axis, then we knocked out, so we can reduce the niche differences. And if this intensifies the interspecific competition, we would expect to see that nitrogen addition actually increase competitive ability differences. And the influence of nitrogen can also throw their influence on the links or relationships between functional traits and coexistence. And similarly, we have fungicide application or foliar fungal removal. So by removal natural enemies, we might reduce niche differences because pathogens, they are one of the drivers to cause niche differences. And some species, they may really benefit from enemy release, so they would really take advantage and then lead to higher competitive ability difference in the community. Again, similar effect could happen on fungicide or foliar fungal removal by influence the relationships between traits and coexistence. So from this slide, I want to show that this framework could be influenced by different global change factors. And another question is that can we actually extend the diversity effect on functioning, on productivity to multifunctions by extend this work to multifunctionality? And here I set one work did by Oscar, and he actually found that niche difference can lead to higher biomass production and decomposition. So this means niche difference can influence multiple functions. So based on this, I would also expect that niche difference will lead to higher multifunctionality. So I need to repeat this again, that species, if they are niche differentiated, so they could therefore differ in their productivity and their influence on energy flow to other trophical levels and nutrient cycling, and therefore have this positive link. And as I mentioned earlier, if competitive dominance by high-performing species or species that can support most functions, we could also see this link, that competitive ability difference positively linked to multifunctionality. But this haven't been fully tested yet. And to test these research questions, actually in Switzerland, we have an IDEA platform showing in the picture here. It's located in Bern, Muchenbergsee, and it's a typical biodiversity ecosystem functioning experiment. And to assemble the plant communities, we selected 20 plant species, and these species range from fast-growing to slow-growing species by seeing they have a gradient of SLA, which is specific leaf area. If the species have per gram of the dry mass can produce larger leaf, which means higher specific leaf area, they are fast-growing species. This is often used in trait-based ecology. And we assembled communities with these 20 species, and we assembled 1 species, 4 species, 8 species, and 20 species. And for each composition of the community, we applied either nitrogen, fungicide, or combined nitrogen and fungicide. Of course, we also have communities which without nitrogen and fungicide. And this is the frequency we applied the nitrogen and fungicide per year, and we weeding the community every year 3 times by removing the colonizers from outside of the communities we assembled. And we also harvest plant above ground biomass to have the above ground biomass data every year. And beyond above ground biomass or shoot biomass production, we also measured other functions in these plant communities. And in above ground, we also measured herbivory and pathogen infection level. This was also measured over time for multiple years. And for below ground, we also measured a couple of functions such as root biomass, soil organic matter, soil carbon storage, and 2 soil enzyme activity, soil respiration, and we also set up decomposition experiments to estimate the decomposition rate and use the beta laminar method to estimate the soil feeding activity. So this is a whole set of the function, but I have to say for some below ground functions, we only measured once because it's too much work to maintain the experiment. And with this data set for the above ground biomass data, because it's over year and in multi-species communities, we also sorted biomass per species. So with this data set, I can actually determine the complementarity effect and selection effects as I mentioned. So here is a example for how I determine this. So if there are 4 species in this community, and we have these species grow in their monocultures, which means they grow alone and they grow together. And they have the observed biomass production. And also, if we assume there is no species interaction, they will have equal relative abundance. So we will also, based on their monoculture yields or biomass production, can calculate their expected biomass production. And from observed minus expected, and then we would have the net diversity effects. And then we apply the additive partitioning framework to have the complementarity effect and selection effects. And here I need to mention that, but I don't show the methods here because it's very complex. I even further partitioned the selection effects to the intra and interspecific components. The interspecific selection effects, which is really the dominance effects, which means the dominance by particularly species, and they also influence the relative abundance of other species. And the intraspecific selection effects, which actually more reflect a recovery from density dependence effect. I will tell this more during the results section. And for the multifunctionality, we used a multi-threshold method, and I simply show the method I used here. So we use multi-thresholds. If we set the thresholds for 50 for a specific community, if we measured multiple functions, if each function passes the maximum function, the 50% of the maximum function, it will count. So in this case, 3 out of 5 actually passed this 50% threshold. Then we count it. The multifunctionality would be 3 divided by 5, which is 0.6 for the multifunctionality of this plot. This is the method we used for calculation multifunctionality in communities. And now I have told you that how we measure functions and how we calculate multifunctionality and how we decompose the diversity effects to complementarity and selection effects. And by connecting it with coexistence mechanisms, actually we need another independent experiment to estimate the pairwise species interaction coefficients and intrinsic growth rates. One of the previous legends from the group, Caroline Daniel, during her PhD, she actually transplanted thousands of the pendive species, the seedlings, into the field with a varying density of heterospecifics, which means interspecific competition, and also the density of conspecifics. And also, she grew these species without competition. By this data set, she estimated under different environmental conditions the pairwise competition coefficients and the intrinsic growth rates. And by applying the structuralist approach, I also showing the method very simply here. Also, the calculation is simply, but the theory behind it is more complex than we thought. But I can briefly explain here. So the competition coefficients of these 4 species, they can build this pyramid, this dark green triangle or pyramid. This size means the structuralist niche difference. If it's larger, which means it can allow a large variation of the intrinsic growth rates. And if it can accommodate the variation of intrinsic growth rates, which means it can coexist. And the in-center of this pyramid and the vector to the set of the intrinsic growth rates indicate is the structuralist fitness difference or competitive ability difference. And here in the calculation, I also applied a non-model. So by randomize the species, so for each community, I have the observed niche and competitive ability differences. And then I standardized it with expected non-model or expected communities. If it's higher than expected, this means this community which really has a higher niche difference or fitness difference. And with these methods, it allows me to compare the coexistence mechanisms across communities with different diversity level. So by applying this method, I determined the coexistence mechanisms for each plant communities based on their zone composition. And because we applied this non-model for 20 species community which have all set of the zone species, actually this will cause the same value of the expected and the observed value of coexistence. That's why for the following analysis, I only involved 4 and 8 species communities, which cover the majority of the plots in our field. So in the results part, as I mentioned before, we did observe positive net diversity effects on productivity, and this was contributed by positive complementarity effects and positive dominance effects or selection effects. And here is the intraspecific selection effects I mentioned earlier, which reflects the recovery from negative density dependency. And it's often indicated as the more negative values of these intraspecific selection effects, the more it's recovered from being in a mixture communities. And here we found that in 8 species communities, actually nitrogen increased the net effect. And nitrogen also surprisingly increased the complementarity effects in 8 species communities and only reduced it in 4 species communities. And the earlier evidence is actually they found, or what we expect is nitrogen just reduce complementarity effect. And here we found that it somehow also depends on the species pool. And here is consistent as we found from other evidence that nitrogen increased overall the dominance effect or interspecific selection effects. And as you see here is that the effect is more nitrogen related. Either we observed the direct effect of nitrogen or the interaction between nitrogen and richness. And I actually detected the previous framework by only comparing the structural equation model with and without nitrogen to compare the path of whether the path is there or not, or modified by nitrogen. And we grouped the fungicide as the variable inside the model. And here, to test the framework before I introduced, we actually found that no matter nitrogen adding or not, we actually found that greater niche difference will lead to higher complementarity effect, and competitive ability difference will lead to higher selection effects or dominance effects. And we also found that the higher niche difference will lead to more recovery from the density dependence effects, because if this is more negative, which means more recovery from being a mixture community. And also we observed that this green line actually it's the path under the control treatment. The blue is the path under the nitrogen treatment. And actually under nitrogen treatment, higher niche difference lead to lower dominance effects, which is more close what we expect. But under control condition without nitrogen application, which a bit old than we expect. And I suppose this is because some short species, which like Plantago, they contribute higher niche differences, but their dominance was limited by adding nitrogen. because some fast species, they can going up and shade the light and probably reduce their dominance. That's why we only see a negative path under nitrogen. And I also forgot to mention, but here I show that the solid line is a positive effect, the dashed line is a negative effect. And we also found that under control, which as expected, indirect interaction does lead to higher niche differences. And nitrogen actually shifted this to negative. And this could indirectly reduce complementarity effects. And we also observed that indirect interactions actually also directly link to intraspecific selection effects, which means with the intensification, with the intensify of the indirect interactions, the intraspecific selection effects are close to zero. And I don't have a good answer for this part to explain why this happens, but I think we need more studies to test why some species it's actually benefited more from being a community where there has less or weaker indirect interactions. And as we expect, we do find that no matter adding nitrogen, fast-growing communities which have lower niche differences and only under control, and we found that fast-growing communities also have higher competitive ability difference. These two paths actually is same as we expect. And we found actually nitrogen shifted this effect of functional composition on competitive ability difference. It's because after adding nitrogen, some of the slow-growing species, their competitive ability difference was increased. And in our system, it's actually, after adding nitrogen, some of the slow-growing species, they are dominating more in the communities. And we also found that functional diversity does lead to higher niche differences as we expect under control conditions. And by adding nitrogen actually removed these parts. And there are also direct effect of functional traits. So we observed that fast-growing communities, there are more intraspecific selection effects happens, which means in fast-growing communities, so these fast species, they may susceptible for herbivores or for pathogens. That's why when they, from being a mixture settings, they recover more. So they have more negative values of these intraspecific selection effects. And actually nitrogen intensifies this. And we also found that only under nitrogen that functional diversity also increased the dominance effect or typical interspecific selection effect directly. And here I need to say that the coexistence mechanism based on the data of the phytometer experiments did for 1 year, but it can still capture the diversity effects, although it's do not fully captured the effects of the functional traits, which means we still see the direct effect of traits on diversity effects. And this model has been complex enough. That's why I think to simply also good for the audience, I would just show other paths in ones with gray lines, and then I don't dive into them too much. So, because later I will also introduce the results about multifunctionality, I think it's nicer to summarize it a bit before we go in that. So actually we does confirmed from our research about this framework, but we do find the links between coexistence and diversity effects, which even consistent was not significantly influenced by nitrogen. But we does see that the major effect of nitrogen to dampen the relationship between functional traits and coexistence, which will potentially indirectly influence diversity effects. Now we are going to move to the results about multifunctionality. And because for multifunctionality, the results was partly published, so I use published results to show you some results from multifunctionality. And for multifunctionality, it's did by another legend from Eric's group, who is Noemi Pichen. And she found that functional composition have positive effects on multifunctionality and nitrogen addition as well. But species diversity effect is not significant, although species diversity effect do interact with functional composition. And this path is very interesting because if we plotted the predicted model, we found that actually the higher diversity communities has higher multifunctionality, especially for these fast-growing communities. So, which means by having this fast-growing species, actually multifunctionality was increased. And I remember our explanation on this result is that these fast species, they may have unique functions. So they don't provide a lot of functions at the same time. But if a community having some of these fast-growing species, because they have unique functions, they will also bring their overall multifunctionality higher. So I would like to extend this framework to link the traits and the coexistence as well. So this is some of the work I'm doing recently. I also would like to show you the results. So here, the dark lines also shows the paths which are consistent or not influenced by nitrogen. So it's consistent between nitrogen and control. And only I use the green line to show the paths exist in control, and the blue line use it to show the nitrogen effect. And we still found that niche difference can lead to higher multifunctionality. And functional diversity can reduce the multifunctionality. And if I go back to her results, the functional diversity effects was here. It was non-significant. And here also because I used just part of the functional diversity level, because I only have 4 and 8 species communities, but she has 1 species communities and 20s. In 1 species communities, this functional diversity, which we measured mean pairwise distance, which is actually 0. That's why maybe we have a different effects here. And again, I found that the nitrogen which are influence or dampen the relationship between functional composition, functional diversity, and coexistence mechanisms, and here mainly about the links with competitive ability difference. And as I mentioned earlier, because we calculate this multifunctionality index across different thresholds, and here is the results only at 1 threshold. And I also show this path, how this path varies across different thresholds. So it is with this figure and the 4 bars, which means 4 different treatment, and the blue and black is same as I show in this structural equation model. And we also have fungicide treatment and fungicide plus nitrogen treatment. But to be simplify this structural equation model, I didn't show the fungicide effect and combined effect by fungicide and nitrogen. So we found that at lower threshold level, this path exists and consistent across treatment actually. But at higher threshold of multifunctionality, actually we only see that this path was exist under nitrogen addition, which could reflect that some plant communities, if we assembled with higher niche differences, they actually are able to utilize the nitrogen addition and take the advantage to build higher multifunctionality. But this I need to further check whether the changes of the coexistence does positively correlated with changes of the multifunctionality. And we also measured functions, as I mentioned, some are above ground, some are below ground. So I checked the cascading effect for 3 above ground functions as well, which are above ground biomass, herbivory, and pathogen infection. And here I only show you the data about the net effect. And we found that actually niche difference, no matter nitrogen or not, it leads to higher above ground biomass, and which cascading to higher pathogen infection. And competitive ability difference, it can reduce the herbivory. And as I explored another functional traits before, which I found that actually in our system, some herbs, which they have higher LDMC, which are dry matter content, it's a defense functional traits. So it's, if having this species, it can have higher competitive ability difference, which may explain why higher competitive ability difference reduce herbivory. It might because this associational resistance at community level. And we also observed that richness reduce indirect interactions, and indirect interactions, same as before, increase niche difference. And richness also have a dilution effect on pathogen infection, but fast-growing communities, they have more pathogen infection, while they have less herbivory. And also we found some effects are only appear in control or nitrogen. For example, niche difference only under control treatment, it's increased the herbivory and reduced the pathogen infection, but this was not exist under nitrogen treatment. And we also found that nitrogen treatment actually have this path where normally if we have more above ground biomass, we will have more pathogen infection. But under nitrogen, more biomass production also means less herbivory. And this could because when we add more nitrogen, the increase of the above ground biomass lead to more pathogen infection. And this increase in pathogen infection actually they reduced the herbivory level because some herbivores, the insects, they don't like to eat the pathogen infected leaves. And also we observed that higher functional diversity under control treatment, it actually reduces the pathogen infection. And some paths are same as before, which is nitrogen also changed the links between coexistence and functional traits. And to sum up these results, I would say that actually our framework can also expand to multifunctionality, and higher niche difference does lead to higher multifunctionality. And this relationship between traits and coexistence are actually contact dependent, and they could potentially influence the effect, or indirectly influence the functions of the ecosystem. And as I'm still working on this part of the project, and later I would like to expand this cascading effect to below ground functions as well. And also we actually measured once the fungal community on the leaves in each community, and also we have some insect data. I would like to check the multitrophic diversity as well if possible. And as I mentioned earlier, functional traits like LDMC or other traits, they may also important. I may consider to complex the model again by adding more traits. And I think this is all of my results, and this is all of my study during my postdoc. And I'm really happy to have the chance to share my research with you. And at the end, I would like to thank Oscar for having me here. And also thank our Pandiv team to maintain this fantastic diversity experiment. I'm sure there will be a lot of questions. Thanks a lot.
Okay, thank you, Xiangyu. Do we have questions for Xiangyu?
Hi, thank you very much for the really interesting talk. Very complex and very, a lot of results, like a really interesting experiment. And so I have a question mainly about the functional composition and how you define it, and whether the definition makes a difference in your potential and your results. Just because you define it mainly on one axis, which is fast to slow growth. But is there any type of variation not explained by that? For instance, reproduction or something. Like, I mean, do they have some sort of, is there maybe a secondary axis that could explain some, you know, inconsistency or results and differences? And I'm asking this because in like life history theory, for instance, you always have a secondary axis that is, you know, that's for instance, like if they have a seed bank, or if they reproduce several times instead of just once, and these types of things.
Yes, exactly. Actually, we start with this fast-slow axis. It's mainly because these experiments, these species were selected actually based on this fast-slow axis. That's why we always use them. But actually, I checked other functional traits, like I mentioned earlier, LDMC, which looks like more or less orthogonal with SLA. And also last year we measured some root traits. I'm sure there are also excess in below ground traits. That's why in the very last slide I mentioned maybe I need to consider about other functional traits as well. Yeah, thanks for the question.
Okay, thank you.
Thank you. Welcome. You mentioned that you remove colonizers.
Yes.
Makes sense, although obviously you don't want to mix more the species in there, but I'm just curious to know whether you collect information about those colonizers and how the rate of colonization might itself be affected by the nitrogen inputs and the fungicide inputs.
Yeah. I go back to the design of the experiment. Maybe it's still very small to see, but actually during the weeding to maintain the experiments, we didn't count which weeds are invaded into the plots. But the good thing is that in our 2 meter by 2 meter plots, we have a strap which is 50 centimeter multiplied 2 meter. We have this strap. We actually left it invasion. We let the invasion happen, and we also track over the time how different species they come. But this work haven't been published. It's one of Eric's work. Perhaps next time when he visited here, she could present this work. But we does check how treatment, initial functional composition and the diversity of the plots, how does this influence the colonization, the colonizers, and how does this modify their diversity effects.
Cool, great. Yeah, look
Thanks.
forward to that. Thank you.
If no one else wants, I have a follow-up question. So this is a multi-year experiment, right? So and of course you manipulate nitrogen, and you have your fungicide. So you have these two treatments. But there is of course background environmental change happening in the system, right? I guess since it's in what is this, in Germany? Yes. No, in Switzerland. Sorry. Sorry, no, in Switzerland, because I saw München, and never mind, sorry. In Switzerland, there has been like recent droughts and stuff. So have you been able to sort of detect some kind of signal of these background stuff?
Thanks for the very nice question. Actually, we do have some sensors we actually within this field site, we measured some local or some microclimate, but I haven't looked into the data yet. And I did once. I looked the drought index, which is a SPEI index. I used the local climate data, and I calculate this for every year, and I correlate with our productivity. And we do found that over some dry years, it's really reduced the productivity above ground biomass. So perhaps because this one, because already complex enough, that's why I didn't to check the over time variation of the diversity effects. But if someone do that work, they should check the environmental climate data as well.
Yeah, I think also would be interesting to see what the interaction between your actual treatments and how, whether this changes some of the arrows, of the effects that you were seeing. I guess,
Hi. Thanks for the talk. I was wondering, as I understand your conceptual model, the coexistence mechanism that is in the background always is competition, right? Either you have niche complementarity, meaning that more species are better for the functioning because they compete less, because the niche sorting, or we have also the sampling effect, which also invoke the competition. And I was wondering whether we can have other coexistence mechanisms in these systems beyond competition, for example, facilitation. Especially I'm thinking in the multi-trophic communities, which is a future avenue, as you show us in the last slide. In that case, will become very evident because as more, for example, if you have more pollinators in the communities, there is more chances for helping some species, helping others, animals, helping plants, therefore more facilitation, therefore more functioning in total. But maybe we also have facilitation in these autotrophic communities.
Actually, I always considered the facilitation between plant species. And actually, when I consider facilitation in this competition matrix, if I keep this positive or facilitation interactions, actually it's blurred or cancelled even the positive link between niche difference and complementarity effects. This is because some fast growing species, they receive a lot of facilitation, but they are just increased the community, the niche difference, but it's do not lead to higher functioning. These fast species, they just contribute very few to the complementarity effects because they are rare. They just trying to hide in the community without distinct. For the point you mentioned, we consider more about the multi-trophic diversity, the interactions between plants and multi-trophic level. I haven't think about this, but once I heard a talk, someone was accessing I think he also called that niche difference by considering the trophic level diversity. I remember his work also found that this can also lead to higher functioning. So it's met, both are exist, but the problem is how to combine all this information together. But for that part, I'm not super familiar, but good that you mentioned. Thanks.
Looking forward to hear more in the future.
Thanks.
Okay, so if we do not have more questions to Xiangyu, please thank him again.
Thank you.